A good day to talk about sex determination. We've talked about genetic sex determination a bit through the semester. Today we're going to focus on it in detail. We'll conclude on Monday with sex determination, and that's all of the new material for the semester. Today, Monday, next Wednesday's a review session. So the essential question is, how do you get males and females? We've got male and female three-spined stickleback fish. How does an oocyte and a sperm, when they fertilize, how does that single-cell embryo know what sex it's supposed to be? Some of it has to do with genetics, some of it not. So these are the goals for today and then also for Monday. We'll get through a little bit of this today, is to look at how sex determination works in three different groups of organisms: fruit flies, Caenorhabditis, nematodes, and then in mammals, focusing on humans. And then the really critical part about this is to understand and be able to describe how mutants taught geneticists about how sex determination works. So mutants that are mutant for sex determination genes, for example. And the reason that this is a broadly interesting question, and I hope it's interesting to most of you, or if not all of you, is because sex determination touches on a lot of different components of biology. So if you're a biology major, hopefully at least some of this is interesting. Because sex determination, how an embryo at this one cell stage starts figuring out whether or not it's supposed to be male or female or not, is impacting basically every aspect of life. Whether or not an embryo decides to develop as a male or female affects morphology, obviously. Males and females look different. It affects behavior. So it affects how these individuals will act if they develop as a male or a female or an intersex individual. Because behavior is involved, that suggests that it also affects physiology. So whether or not you're a male or a female or intersex has something to do with your physiology, hormone production, for example. How your cells respond to the hormones you produce. So pretty much every aspect of biology is influenced by what sex we are. This is bizarrely crazy because what I've tried to argue just now is that sex is the most fundamental decision that you made when you were growing up. And every individual of every species, what sex you become is the most important decision. It affects morphology, physiology, behavior. You'd kind of expect that maybe if figuring out which sex you are is that important, biologically speaking, that the way that we figure out, the way that individuals decide what sex they're supposed to become would probably be something that's pretty stable. It's not something that you'd want to have mutations occur in. It's not a process that you'd want to be plastic, something that's easily influenced. So what happens, this is an evolutionary context now, so don't freak out. You'll get this again when you go to Biology 105. Why is sex determination so critical from an evolutionary perspective? Sir?
Oh, sorry.
I know you're waving at a fly, but that's okay. Yeah, it's the end of the semester, so the flies have come out. Yeah, lots of flies. Don't sit in the front. That's where all the flies are. It's not me, I swear. What's the problem with mutations in sex determination? If there are genes that cause you to be either male or female, what happens if you get a mutation in one of those? What could happen? So one of the possible outcomes is that your body doesn't pick a sex, so you might be a hermaphrodite or an intersex individual. Sometimes that leads to sterility. What happens if you're sterile? You don't have kids. So you'd expect, and this is just a basic genetics and an evolutionary principle, you'd expect that every species that uses genes to figure out whether or not they're supposed to be male or female would not want to have mutations occur in that process. Because a mutation is going to affect what sex you're going to be. It affects whether or not you can have kids and pass your genome on to the next generation. So what this slide indicates is that that's actually not the pattern that we see. it is when you get in really close to looking at small groups of species, but across the tree of life, across multicellular organisms, in this case vertebrates, there's a lot of different ways that organisms figure out how they're going to become male or female. So for example, here the squares, this is a phylogenetic tree indicating a bunch of different taxa that are vertebrates. Where are we? Humans. We're not marsupials. We're close to marsupials, though. We're eutherians. So we're eutherian mammals. So the way this is drawn, we are represented at the top of this phylogenetic tree. So the tip of each of these branches represents present time, t equals present. So there are individuals, there are lots of mammals that live at this day and age. There are lots of marsupials, there are monotremes. Do you know what monotremes? There's one. It's the weird mammal. Platypus. So a monotreme is a platypus. Right? We got snakes, lizards, the tuatara, birds, crocodiles, etc. The squares and circles and diamonds tell us what sort of sex determination mechanisms each of those groups of organisms uses to determine sex. So the squares are situations where the XX individuals are female. Individuals that have a Y chromosome are male. That's what the key at the top left says. I'm just redrawing it because I'm about to zoom in. The circles represent the opposite, where if you have two of the same sex chromosome, that makes you male, and it's the individuals that have a W chromosome that turn into females. The diamonds indicate environmental sex determination. So there's our vertebrate tree of life. What sort of sex determination do mammals use? What is this? What do you know about it? Don't even look at the figure. What sort of sex determination do we use? Do we use genes that tell us what sex to become, or do we use our environment? We use genes. Okay. So humans have that XX, XY sex determination system. So does every other mammalian species that we know of. So every single mammal has a Y chromosome. That's what tells males to be males. Okay. Same is true in marsupials, of species that have been studied. So this doesn't necessarily mean that there aren't some weird, rare species out there that don't use these approaches, but this is the trend. Okay. Then we get into snakes, which have this ZZZW sex chromosome system. Okay, then what happens when we get to lizards? What sex determination system do different lizard species use? Everything! Different lizard species, this is not one species, by the way. This is a tabulation across all lizard species. Some species have XX, XY sex chromosomes. Different species have ZZ, ZW sex chromosome systems. Some species use environmental cues to tell them what sex to be. This freaks evolutionary geneticists out because what we expect is that if once an organism or a group of organisms figures out how to figure out what sex to develop as, you wouldn't want that process to be tinkered with. But there's so much plasticity here. You could look at two different lizard species, and they use totally different mechanisms to figure out what sex to develop as. But then we get to birds. One sex determination system for the whole group of birds, every bird species. If you get a W chromosome, you're a female. Right? That's ZW. The two sex chromosomes, like XY in human. And if you have no W chromosomes, then you're a male. Sorry, if you have no ZW chromosome, you're ZZ. And you're male. So, thank you. So lizards and birds I've come up with two different versions of the same approach, using a female-specific chromosome that determines femaleness. And sort of the default sex, if there is one, is male. Without a W chromosome, you're a male. You add in a W chromosome, then you become female. And then you look down farther here, and we see that both turtles and fish also have this great variety of sex determination mechanisms. So you can find fish species that have ZW chromosomes, that have XY sex chromosomes, and that have environmental sex determination. I've mentioned environment. All right. So the very first clues that we got about how this works were in 1905. Boring. Don't remember the year. Doesn't matter. When microscopy got to the point where you could actually look inside cells, as we talked about at the start of the semester, and actually view chromosomes of metaphase. And I've probably shown you this slide before even, where you can look in Drosophila cells and see that males have a specific shape of a chromosome that doesn't occur in female cells. So there's one chromosome there in males that's kind of a hook shape or a little bent, like a letter V. The Y chromosome. And the way that the first species, Drosophila melanogaster's sex determination mechanism was figured out was purely correlation. So every time a scientist looked at a female set of chromosomes, or a set of chromosomes for a female, they had two chromosomes that were the same shape. So every female had chromosomes that looked like that. And they happened to look like that. And males, every time they looked at a male, didn't matter which male, pick a random male out of their population, there was one chromosome that looked like that, and then there was a smaller chromosome. So how did the sex chromosomes get their names? Why are these called X chromosomes? It's not because they look like X's. We didn't even know that chromosomes existed back then. So the person that saw these first, chromosome, right, colored body, they stained these chromosomes so they could see them, and they saw that there was one thing that always correlated with sex. And that became the X factor, the unknown factor. Nobody knew what these were. They didn't know about genes. They didn't know about deoxyribonucleic acid. It was something they could see in the cells that had a perfect correlation with the phenotype of sex. And why is this called a Y chromosome? Why not? The next letter after X. It's not because they look like X's. That's not the case. It was just the X factor, the unknown factor that seemed to have something to do with sex determination. So how would you experimentally demonstrate if you thought that a species was using a chromosome or a gene to determine sex? How would you prove that that was actually the case? So how would a Drosophila geneticist prove that these chromosomal differences that they saw were actually causing, so there's a genotype, we know now, these are chromosomes that carry genes. How would you prove that those genotype differences are what cause these phenotype differences? What sort of experimental manipulations can you do?
Like sufficiency and necessity?
Right, so this gets back to the concepts of necessity and sufficiency. Starting to draw material back from earlier. So let's think about that for a second. Here's what we know. In Drosophila, if you have an X and a Y chromosome, you're male. If you have two X's, you're a female. In Drosophila, it happens to be the number of X chromosomes that dictates sex. So there are two genetic differences between females and males. We've got the number of X chromosomes, and we've got the number of Y chromosomes. So females have two X's, males have one. Females have 0 Y chromosomes, males have 1. So it could be either of those two things that causes sex determination. It could be that Drosophila cells know how to count how many X chromosomes they have. And the number of X chromosomes tells you whether you're supposed to be male or female, or it could be that they know whether or not there's a Y chromosome present. That's a critical point. Now's a good place to ask any clarifying questions if you have any. There are two genetic differences between these two sexes. Number of X chromosomes and number of Y chromosomes. In a Drosophila, it turns out that the way that individuals figure out their sex is they count how many X chromosomes they have. It doesn't have anything to do with the Y chromosome. So what sort of mutants would we want to be able to demonstrate necessity or sufficiency in sex determination? It's not going to be XX, it's not going to be XY. Those are the majority of individuals. What were the mutants that people identified in Drosophila, do you think, that helped them figure out that it was the X chromosomes and not the Y chromosomes that were important? So you want mutant flies that say lack one of the chromosomes. So what would you do? Have a fly with no sex chromosomes? They're not viable. So you can't have nothing. So what's the next best thing? Pardon?
Add a Y to the double X.
Okay, so you could look for XXY flies. So according to this chart over here, they'd have what? 2 X chromosomes and 1 Y chromosome. Which of these does this test? Is this a necessity test or a sufficiency test? We're asking about the Y chromosome here. So if you added a Y chromosome to an XX individual used to be female, what happens if that individual is male? Tells that the Y chromosome is sufficient for maleness. If, and this is, we don't know this yet, we're just setting up the experiment. If the XXY fly is male. So if you take an XX, a female fly, and all you do is add a Y chromosome, and that turns her into a him, that means the Y chromosome is carrying some genetic factor, a gene or genes, that cause maleness. Female to male. What sort of a genotype would you want to test the sufficiency of a Y chromosome to make a male? Sorry, the necessity. Sufficiency. What about necessity? What do you do if you want to know if the Y chromosome is necessary to be a male? Get rid of it. So that makes you what genotype? X or XO. So geneticists usually write in an O, which means nothing, 0. It's not a 0 though, we call it XO, not X0. Go figure. 1 X chromosome, no Y chromosome. So if that individual's female, that suggests that the Y chromosome is necessary to produce males. So those are the types of mutants you'd look for if you wanted to understand which of these processes was controlling sex. Is it counting X chromosomes, or is it the presence or absence of a Y chromosome? So I'll leave it to you to think about how this same set of mutants, XXY and XO mutants, can also tell you about whether or not X chromosome counting in this case occurs. You can use these exact same mutants not to test for necessity and sufficiency of the Y chromosome, but also necessity and sufficiency of two X chromosomes to cause females. Questions or concerns? I'm not going to go through that in detail because it sounded, as I said it, like a good exam question. So here's where we're at in Drosophila and in humans. This figure is exactly the same for Drosophila and for humans. Right? Your XX, your female. If your XY, your male. And the really critical part about all this is that with either X chromosome counting, two X chromosomes versus one, male, female, or presence or absence of the Y chromosome, the really important result is that you make an equal number of males and females in the population. So you get a 50-50 sex ratio. Half of the offspring get a Y chromosome, as we've seen in our pedigrees, half don't. You expect in humans and in all these other species that do genetic sex determination, a 50-50 sex ratio. Half males, half females. It means you don't go extinct. Because you've always got members of both sexes around to mate with each other, even if they happen to be brothers and sisters. In extremely serious situations, you could still reproduce. What happens in environmental sex determining systems? How might environmental sex determination be sort of disadvantageous?
Well, if they all incubated at the same temperature, then you have all the same sex.
Okay, so if all of the eggs incubate at the same temperature, imagine you're a turtle. You go up on the beach, you lay all your eggs, cover them with sand. Imagine they all incubate at the same temperature. Then you produce entirely one sex of offspring. Does that seem like useful for the maintenance of your lineage? No. Fortunately, some other turtles somewhere else laid eggs at a different temperature and they produced members of the opposite sex, and then out in the ocean, maybe someday, they meet and they mate and they reproduce in rainbows, and everybody's happy. What happens when global warming happens? And everything's hotter, and so that temperature passes the threshold where you no longer anywhere on the planet make individuals of one of the sexes. All of a sudden, temperature-dependent sex determination seems like not such a great approach to figuring out what sex to become. Everybody everywhere is all one sex, and then you're extinct because there's no more reproduction. This is slightly different in my nematodes, C. elegans, C. briggsae. I've told you before, and these are just pretty pictures. There are hermaphrodites and there are males. We can consider them females and males for the sake of being in parallel structure with what we've drawn up here for Drosophila. So it turns out in C. elegans and in my favorite, C. briggsae, the same mechanism of X chromosome counting that happens in Drosophila also happens in this species. If you have two X chromosomes, you're a female or hermaphrodite. And if you have one, you're a male. So it turns out that there are no Y chromosomes in C. rhabditis, so the males are naturally XO. They just have one X chromosome and that's it. No one even had to bother with the necessity and sufficiency experiments because there was no Y chromosome to lose. So there's only one option. It must be the number of X chromosomes that dictates sex in this species. Technically a hermaphrodite. So, two X chromosomes, you're a hermaphrodite. One X chromosome, you have this genotype, you're a male. Now we get to the point where you get to do something, briefly. This is the genetic pathway that controls sex determination. So these are the proteins that are involved in rhabditis, elegans, or briggsae in sex determination. This should look pretty familiar. We've gone through some examples like this before in class. Let's see. Let's work through this. What are these wavy lines? What do you think? My eyesight must be going bad. It looks like DNA. That's the nucleus. Okay. Tra1 is a transcription factor. So it can normally go inside the nucleus and it turns on genes that are important for making hermaphrodites. Tra1 protein, the transcription factor, binds to DNA and it turns on, it activates transcription, makes genes that are important for actual development of hermaphrodites. And an XX organism is which sex? That's the hermaphrodite. So this is what you want. If in an XX organism you want Tra1 in the nucleus transcribing genes that make you a hermaphrodite. That's the goal. And the way this works is that there's a gene that produces a protein called a fem protein, or feminizing protein. And if fem gets a hold of Tra1 as it does over here in males, fem prevents Tra1 from going into the nucleus and transcribing a gene. But then there's this gene and its protein product Tra2, whose job it is to grab onto the fem protein and prevent it from doing its normal job. And then, in males, they produce, if you don't have two X chromosomes that is, if you only have one, those organisms, those individuals produce a protein called HER1, hermaphrodite 1, whose job it is to prevent Tra2 from grabbing onto them. It's a genetic pathway. So what I'd like you to do is to draw out this genetic pathway using the blunt arrows and the straight arrows that we've talked about. I'll give you a few minutes to work on it. This slide is probably worth using. Just draw that pathway up here in the top. Your pathway should start with the two different sexes. So you can imagine having one pathway for exo organisms. So this is how I would start. Exo organisms produce the HER protein. And then how does that connect into the rest of this cascade, these different genes and proteins? Is it a solid arrow? Is it a blunt arrow? And go from there. And then I'll launch a Google Classroom assignment now to attach this to.